students. In today's class, we will learn about the different sequencing technologies, particularly the first and second generation sequencing technology, how they are done, how they evolved, and their advantages, their limitations. Okay, so let's start with some common things that we hear in sequencing. So what do you exactly mean by sequencing of microbes or sequencing of eukaryote or anything that you want to sequence? So basically what it means is like we have to determine the identity that, for example, this is a sequence where just a second. Okay, so these are the different characters in a DNA sequence. So we have to identify that, okay, at position 1 A is present, at position 2 T is present. So basically we are trying to see what is present at what position. So both identity and their relative position is important to know. Okay, so this is called sequencing because it is also a sequence of DNA, sequence in which the 4 nucleotides A, T, G and C are written. Okay, so it is mostly done for DNA, but sometimes RNA, right? To see the history of these sequencing technologies, basically it evolved with the Sanger sequencing when Sanger gave this method in 1977. Very small bacteriophage and small microbes were sequenced initially, followed by the instrument that came, ABI sequencer, and then the pyrosequencing, and so basically till here it is called like first generation sequencing. Then there came second generation, third generation and fourth generation. So today what we have is advanced second generation and the fourth generation technologies, and of course Sanger will stay because of its own advantages that we'll see today. Okay, so with after Sanger it was pyrosequencing and 454 instrument that came, which were very promising but suffered because it was a little expensive to run. The sequencing cost was higher. Followed by Illumina sequencer and a parallel technology called solid. However, among the two, among the solid, ion torrent and Illumina, basically Illumina had better chances of survival, and still it is very commonly used today. And recently we have PacBio, Nanopore and 10x Genomics. So basically these are, you can be said as the third generation technologies. So just to give an overview of the different first, second and third sequencing technology, you can see that the first one basically used to give, so okay, it was based on Sanger. Maxim Gilbert was also in method, but basically the instrument that was developed was based on the Sanger sequencing technology because it was more efficient and easier to do, right? So it gives long read length of almost like 800 base pair, right? Low error rates, which is good. And the instrument can be run for a smaller fragment also, or a single fragment also. Okay, then the second generation sequencing are massively parallel. So here we do not go for a single fragment but for like several millions of fragments together. Okay, and but their disadvantage is that they produce like shorter read and basically sometimes PCR biasness, but speed is very high and it is very high throughput. And this is the method that has been commonly used for most of the sequencing projects and metagenomic projects that has happened so far. And now what we have is the third generation technology where the read length is very long, but instrument is expensive, technology is still expensive, but it is very promising and gives very long read length, and which is important because otherwise you spend a lot of time in assembling the sequences, the shorter fragments, and it also has its own like errors and assembly errors and those kind of things. So we will also see like what third generation sequencing technology is in the later classes. So today we will cover the first and second generation sequencing technology. So in genome sequencing, the major challenge is that there are so many genomes that are available for sequencing, and their length is not a few hundred or thousand base pairs, but millions and billions. For example, a bacteria is 4 million base pair, whereas a eukaryote is half GB or half billion to like several billion, 3 to 4 billion base pairs also. Like for example, in case of aloe vera plant, it is 16 billion base pairs, so which is very, very long. So we cannot use, of course, the first generation sequencing technology, which gives only 800 base pair, and we have to use massively parallel or contiguous sequencing assemblies technologies. Okay, so third generation is recently come, which is very good, but most of these genomes have been done by the second generation sequencing technology. And the common terms that we see in sequencing is, say, shotgun sequencing. So basically the DNA is very long. For example, this is a bacterial circular chromosome, which is, say, 4 million base pair. Okay, so we cannot sequence 4 million at one go. So we have to break this into like smaller pieces, and those pieces go into the sequencer. So that is called shotgun. So we break the DNA. We will also see this in the next slide. Then when we break something into smaller pieces, we again have to reconstruct or reassemble those, so that we can again from these smaller pieces we stitch together and we get the complete circular genome, or a linear chromosome in case of eukaryotes back. Okay, then when we say like we break that into smaller piece, so basically the smaller piece which comes out of the sequencer, or basically which a sequencer can sequence or read. Okay, so it's a DNA piece and the sequencer is reading the piece and telling you that okay, what base pairs it is composed of and what is their relative position. So this is called a read. So it is reading the DNA from a single piece, right? So it is read, which can be both paired end and single end. Then when you have reads, you have to put those reads back together, and for example, this is one read, this is another read, so you have to put that back together looking at what is common between the end of the first one and the start of the another one, and if both are same, then basically we can connect two together and make a longer fragment. So this way we keep on connecting the reads together based on the overlap at their ends, and this is called an contig. Okay, so what we are doing here is we are taking a chromosome, breaking into pieces and then trying to assemble the pieces back. But in this process, it is not always guaranteed that you will get the complete circular or a linear chromosome back. So there will always be some missing gaps in between. So that is why the first sequences that come from for a genome are not a complete chromosome, but a draft sequence or a draft genome, right? And when we have to make more efforts in completing that genome. For example, in case of human genome, it was started in late 90s, and then the first draft came in year 2000, and then the complete chromosome or complete the telomere to telomere of the complete chromosomes of human genome came last year. So it took almost like 25 or more years to complete the human genome. So we start with draft sequences and then we reach to the complete genomes. And based on the various insert sizes, and so scaffolding is a process by which we can even further build the contigs together into longer pieces. Okay, and actually these scaffolds help in constructing the draft sequence. Then a read can be a single end and paired end. For example, here you can see, so if this is a piece of DNA, and if we read only from this side, it is called a single end read. Whereas the same piece of DNA, if I read from both sides, the okay, forward and reverse, okay, so we generate R1 and R2 from here, forward and reverse read. So basically it is called a paired end sequencing, right? And for example, if the piece is say 1400 base pair, okay, so from Sanger, for example, we can read 800 base pair, right? And from the other side we can read 800 base pair. Okay, so there will be an overlap, some region which is covered both by the forward read. This is the forward read and this is the reverse read. Okay, and together they are called the paired end read. So this way when we can assemble these two reads together into a single piece. Okay, and there will be some region which was like common between the two. Okay, and that was used to stitch the two reads together. Okay, so this way when we keep on stitching in both directions, and this becomes a contig. Okay, so single end and paired end. And then ESTs were smaller sequences, that because initially the sequencing cost was higher, so from a transcriptome, like we used to sequence the smaller pieces, and those pieces were direct evidence of the presence and absence of a transcript or a gene expression product. okay, and also they were very helpful in predicting the intron exon structure and the alternate slicing events. So ESTs were very helpful that way. And then KB is kilo, that is 1000 base pairs, and megabase is 1 million base pairs, and similarly 1 GBP, when you see, it is 1 billion base pairs, 10 to power 9, and million is 10 to power 6, so basically 3 orders more. And locus could be any region on the chromosome, okay, that you want to sequence, so that could be the locus of sequencing. And similarly, like there is a plasmid chromosomal sequencing and plasmid sequencing. So plasmids we have also seen in the second class earlier. So plasmids are extra chromosomal DNA that are present in bacteria, and which contains genes which are important for the additional functions or the function that help in the survival of that bacteria. Okay, so we have done the single and paired end sequencing also. So now various versions of sequencing, as I mentioned, it is completely sequenced, that if I know for a complete bacterial circular genome all the pieces, and we know that okay, it starts with this, or EC gene is here, and we have done the complete sequencing from like start to the end, right? So that is a completely sequenced genome or completely sequenced piece of DNA. Draft version is that there are always like some gaps, like I know this piece, and this piece, but there are 2 gaps in between and 3 pieces. So draft version of genome, generally the genomes are in draft version initially, and later on they are completed. Coverage is a very important, that you have to estimate beforehand that what coverage you want your sequencing to be done. So that means like how many times you want to read the genome, so that you have sufficient number of pieces to assemble them back together into a single genome. Okay, so the formula for example is N into L divided by G. So N is the number of reads, L is the length of reads, okay, coverage equal to, and G is the size of genome. For example, here I have mentioned, for example, to calculate the coverage, so N is number of reads, it's 8, okay, length of each read is say 500, okay, right, and the genome size is 2000 base pairs, okay, so coverage will be is 4000 divided by 2000, so 2. So basically coverage is 2 or 2x. So that means you have read from a number of reads that you have generated, you are able to read the genome 2 times, right? So it is not essential that you are reading from head to tail 2 times, but overall the number of pieces that you have generated, it could be from one end, another end, or covering the entire chromosome basically. So those number of pieces amount to 2 times of the DNA that was present in that chromosome, right, or in the piece of DNA that you are sequencing. So estimation of coverage before and after sequencing is very, very important. Okay, so we saw the term shotgun sequencing. So shotgun basically, you know, like it's a the term means like some kind of gun, which is, and gun is basically you are breaking something into pieces. Okay, so like this is the initial DNA that you have got from a cell. Okay, so you have to fragment the DNA into smaller pieces. Okay, so basically this methodology that I am telling was the typical methodology that was used to you know, sequence the earlier genomes like Drosophila, C. elegans, yeast, or human, right, where the DNA pieces or complete chromosomes were broken into smaller pieces, and then those pieces were cloned into vectors, okay, and then those vectors were transformed into the bacteria. So what we have done here is, so there was a piece, okay, which was cloned into a vector, and okay, so basically why we do this, and basically this goes into a bacterial cell, okay, and so like a plasmid, you know, so whenever this bacteria divides into 2 copies, so now after some time bacteria they will divide, so it will have now 2 copies of that fragment, okay. So now with one division, your piece of DNA which was here, okay, also became 2 copies, okay, and this way the bacteria will divide into colonies into and form a like single pure colony containing like millions of bacterial cells, and each bacterial cell will contain also your choice of DNA, So that means you have basically amplified your single piece that you have cloned in this vector or plasmid, okay, into billions of copies or millions of copies based on the cell division which bacteria has done, and along with that your piece of DNA which was in the form of plasmid, okay, insertion into a plasmid also got amplified, okay. And this is important because like when we break the DNA, we have a single piece from each. For example, if I break this, I'll get a single piece, okay, of DNA. So I have to have sufficient quantity of DNA, amount of DNA for sequencing, okay, and a single piece may not be sufficient for that, okay. Maybe the third and fourth generation technologies can do this, but initially the earlier technologies needed some amount of DNA for sequencing, and this way by cloning them into vectors and amplification by okay, so this way we can increase the quantity of each piece, right. And this was also important because this way we are also making a library of the each pieces that we have got from the genome. And initially these approaches, library construction and cloning and transformation, were very important for before the sequencing, okay. So okay, and this way we will then break this open and get this piece out again. Now we have got, okay, multiple copies of the same piece, like millions of copies maybe, okay, and that is equal to sufficient amount of DNA, that like more than 1 or 2 microgram that we are needed for sequencing, okay. And then those were taken further for sequencing. Okay, so now there are different sequencing platforms, and if I say that how they evolved, for example, this is the typical Sanger sequencer, okay, and this is maybe the sequencing room. You can see like 1, 2, 3, 4, 5, 6, 7, 8, 9, and basically multiple rows of these sequencers, okay, on the different lab benches, okay. So the entire room is full of sequencers. So you can imagine that this was one of the rooms that was used for sequencing the human genome, and these machines run parallelly day and night to sequence the genome. But now with the advance of sequencing technologies, you can replace the entire room by a single machine. For example, I can say like in the years like 2010, Roche was very popular, so you can replace it as entire room by a single Roche machine, okay. And then today you can replace multiple of these Roche machines, or maybe a room full of these Roche machines, by a single Illumina machine, okay. So this way the technologies become massively parallel and high throughput, okay. You can imagine that thousands of such machines you can replace by a single Illumina machine, okay. And the cost is also like several folds lesser, okay. So okay, so we'll also get an idea of cost later, but like these days you can imagine that in 1000 rupees you can get 1 billion okay, 1 GB data, or 1 billion base pairs sequenced, right. So let's start with the most important technology that started basically the concept of sequencing, that was the Sanger sequencing technology. So basically there are DNA replication. So in DNA replication you need a DNA, okay, a primer, okay, dNTPs or nucleotides, okay, for sequencing, okay, and some other mix that is needed for the PCR reaction, okay. But here we are not doing PCR, we are doing sequencing. So basically, and a polymerase, of course we have, this is very important, okay, a DNA polymerase, okay. So these are the 4 essential things, okay, and same things are used in sequencing also. So here basically you have a single piece of DNA, okay, in sufficient number of copies, okay, and that means you have the required amount of DNA that is needed for sequencing, okay. So now you have 4 nucleotides, 4 dNTPs, okay, and for each dNTP you also have a dideoxy dNTP, okay, dideoxynucleotide, okay. For example, here this is a regular nucleotide, okay, right, but here you can see that in place of H you have OH, right. So presence of this actually blocks the polymerase from adding further nucleotides, okay. So wherever this will get inserted, this piece, it will stop the reaction. So that is why it is also called chain termination sequencing. Okay, and okay, so it generates a library of fragments or multiple fragments because of the random insertion of different DNTPs into the sequence. Okay, so it requires a DNA primer, DNTP, DNA polymerase, okay, and a sequencer which generally like which has a which do capillary electrophoresis later to separate those fragments that you have generated. Okay, okay, so okay, so like this generates a decent length of 400 to almost 1000 base pair, and average length that you get always is like 7 to 800 base pair, which is reasonably good to for either for identification of a gene or a molecular marker, right, and very low error rate, and basically it played a very important role in the human genome sequencing. Okay, so now this is the DNA fragment. Okay, so now you need a primer that you will give along with the DNA piece that you are giving for sequencing. Okay, and then consider that you have 4 different flasks. So each flask contain the 4 nucleotides. Okay, all the 4, plus in addition they will also contain 1 dideoxynucleotide. Okay, like this will contain all 4 nucleotides, but also a C dideoxy, a G dideoxy, A dideoxy, and T dideoxy. Okay, so in this, wherever T will occur, okay, so T can occur like A, T, G, C, A, T, C, A, G, T. Okay, so you can get a fragment which is, because whenever T will occur it will block, so A, okay, or it could be A, T, G, C, A, and then the blocker thing came, so another fragment could be this, and it could be A, T, G, A, T, C, A, G, okay, and a blocker. So you'll get 3 fragments from here, and similarly like for G, C, or A or T, you can get different types of fragments. Okay, so this way you will get a you can say ladder of fragments which you will separate by capillary gel electrophoresis, which is sensitive up to 1 base pair also, right. So basically it starts with, it has 4 different steps: denaturation of DNA, because you have to anneal your primer there, okay, with the primer annealing, and then you are extending the bases using the combination of 4 DNTPs and 1 dideoxy, okay, dideoxynucleotide, okay, and these as I mentioned here, like this can get inserted, okay, at various places and gives you a list or library of fragments where these random dideoxys of all 4 different types got inserted at various places, right, which you are separating on this gel. For example, here, like if this is T was inserted here, and like A was inserted here, and like that A, okay. So these are the different dideoxys, and basically it is important to produce these dideoxys by great accuracy, okay, and these are also commercially available now, okay. So okay, so basically you have 4 tubes with 4 regular nucleotides, the normal DNTPs, and 1 DNTP with a blocker, dideoxy DNTP, okay, nucleotide, and basically it can get inserted at various places into the sequence, and when you run that on a gel, you can know that, okay, like this was the T, okay, this was A, and then again A, then this next one is T, then the next one is A, then this one is C, and this way you can continue reading the sequence, okay. So this is the library of or ladder of different fragment that you have generated by random insertion of ATGC, dideoxy ATGC into the sequence, right, and then you are separating that with the capillary gel electrophoresis, and using the laser you are detecting that what is it is A, T, A, G, or C, okay, because actually these dideoxynucleotides are also fluorescently labeled. There are 2 types of rhodamine and fluorescein dye which are used their derivatives are used for the 4 different nucleotides. Okay, and then you separate that into the ATGC DNA chain, right? Okay, and for each position, like it is A was detected here, that means the last dideoxy was A that got inserted at this position. Okay, and it gives a fluorescent signal of green, so you'll get a peak of green. Okay, then T, then G, then C, then T, and this way you can continue reading and building your sequence, and you will also get a peak for each base pair, because these dyes or these dideoxynucleotides were also fluorescently labeled, right? And later on you can always use this image to know that in case you have some confusion about some base pair at some position, you can always use these images, and maybe like at this position you had some confusion and N was there, so you can decide that what base pair should go there. Okay, so as I mentioned, like 2 types of fluorescent dyes are used, fluorescein and rhodamine derivatives for the along with the which are attached to the dideoxynucleotides. Okay, this is a typical sequencer, genetic analyzer by ABI, which is a company. Okay, and you can see these are capillaries here, right? Capillary gel electrophoresis, and 8 capillaries are visible here, and like I have just shown you that image inside the box, right? And today we have a variety of such sequencers for smaller and larger needs, and many labs have these sequencers in house, so that they can routinely do the sequencing, because these are generally needed for everyday requirement, and not very expensive also. Like for example, for one piece of DNA sequencing, one reaction, it will cost like about 300 to 400 rupees, which is reasonable, right? And like different instruments you can see here, like you have different number of capillaries, 4, 24, up to 24, up to 96 capillaries. Okay, which is good, because you can see that the requirements may be different in different labs, and or different places where they are used. Okay, and based on that you can buy the sequencer. And this Sanger method, dideoxy method, is very important. It produces a good amount of DNA, 500 to 700 or up to 800 base pair accurately, right? Even longer you can get. Okay, but so it is, so like this will cost like say 300 rupees. Okay, but so for a single piece of DNA, if you want to use this, it is reasonable. But if you want to do it for a complete genome, like from a 1 million or 2 million base pair bacterial genome, it will be very, very expensive. You can imagine that it will have thousands and thousands of those pieces, and into 300, that will be like you know, multi-million rupees project, so which is very expensive that way. Okay, and the initial technology of shotgun sequencing, like cloning, transformation, and then colony picking, and then you know, PCR also sometimes, so that was very tedious and time consuming. But you know, the latest next generation methods do not have these steps. Okay, and they are independent of cloning and culturing, but Sanger for the genome sequencing was dependent on cloning and culture, you know, culturing of the bacterial cell before sequencing. Okay, and we of course cannot use it for the sequencing of billions of microbes that exist on this planet, or billions of genomes, including, you know, bacteria, fungi, yeast, molds, insects, birds, animals, plants. Okay, indeed humans have done. Okay, so that is not possible by this, but it will always have a role and importance, because at least for the identification of those, when you are using a molecular marker, you cannot use a next generation sequencing technology, but you will be using a Sanger sequencer. Okay, then after this chain termination method, that was the principle of Sanger sequencing, we are now coming to next generation sequencing, which is sequencing by synthesis. And this is also called deep sequencing, because you are generating like, because you are reading several thousands copies of each fragment, okay, and then making the call for the base pair, and you can also sequence the complete genomes within hours. So this is the typical Illumina instruments, and it relies on the sequencing by synthesis technology. So in the Sanger, if you remember, we used to have 4 normal nucleotides and 1 nucleotide with dideoxy. So here also that nucleotide has a blocker at the OH group, okay, but that blocker is reversible. So you can always remove that blocker and continue with the sequencing. So basically what happens is, like, that blocker can get inserted into the DNA, and once you have, like, this free OH end is needed. So if you have a blocker here, so basically the sequencing will pause, okay, but it will not be extended further, this chain of DNA, okay. But if I remove this blocker, then again I can continue with this. So basically you can pause for a moment, you can know that what has been inserted, okay, you can read that. So like for example A was there, then you remove this blocker, and then you can also continue further. So for every base pair you will pause for a second, okay, and remove the blocker, and then again go on with the sequencing. So this is called sequencing by synthesis, right. And this, along with the OH blocker, or basically the it will also contain the a fluorescent dye attached to this nucleotide, which will give the signal for the various 4 nucleotides. Okay, so sequencing by synthesis is one term that we have seen, and it is massively parallel because at one time you can sequence millions of pieces together, right, and you require a blocker DNTP, okay, and also a fluorescently labeled nucleotide, right. And here you will not use like in Sanger you were using 4 normal and 1 blocker. Here all 4 will have a reversible blocker attached, okay. So all nucleotides will have the OH group protected, which will be later on released for further sequencing. Okay, so it basically the technology started with Solexa, which made the first sequencer. It was called Solexa. Now then it was acquired by Illumina, and it became Illumina Solexa, okay. And this is a typical flow cell. You can see a $1 coin. So basically it is kind of glass slide, okay, which has different lanes, you can see here, okay, where DNA goes in and gets sequenced, okay. And then this slide is kept, okay, in front of a laser. So there is a laser excitation, and then it is the signal which comes out of this is read by the camera. Okay, so it has 4 steps. One is since you have broken your DNA into pieces, you want to first of all prepare a library of those pieces. So you will label that with the forward and reverse adapters, and then for each piece, like in Sanger, here also you need multiple copies of each piece of DNA so that you have sufficient quantity of DNA for sequencing. Here it is mainly for the signal that you get from the fluorescence, okay. So you also do the amplification, but that amplification is done on the flow cell, okay. You don't need to do this by PCR or by colony transformation, but it will be done on the slide, okay, or the flow cell, okay, followed by sequencing and then the analysis, okay. For example, this is your DNA. So you break the DNA into smaller pieces, right. So your DNA is broken into smaller pieces. So for each piece, basically at both the ends, okay, you will put adapters, right, forward and reverse, which are so, and this way, okay, and then make multiple copies of these on the flow cell, right, and then each copy will be sequenced, so each piece will be sequenced parallelly, right, and you will get the signal. So we will go in details of this. So first of all, what we have to do is, from the genomic DNA we have to construct a library, okay. So this library construction takes a few hours, and it is done before actually you go to the instrument, that is the sequencer. So once the library is prepared, then you load this library on the flow cell that you have seen the glass slide, okay, and then that flow cell is put in the sequencer and the sequencing happens, okay. So like this is your, and now start with the like how it is done. This is your piece of DNA, and these are two adapters, you can see, like pink and purple for convenience. Okay, so you have put the adapters at both ends of the double stranded DNA, okay, forward and reverse. Okay, so now each piece contains two adapters. So now I know what is there at the start and end of the DNA, because to start with, these were random pieces of DNA. I did not know what is there at the start and end of the DNA, okay, and what is this DNA is about, okay, what is the sequence of it. But now after putting these pieces at the forward and reverse positions, okay, start and end, I know that I have some marker sequence at start and end, and I know like at least the start. I can put this, use this as a to put my primer, also to bind to some surface and like that, which we will see later. Okay, so now you see that we have put adapters at both the forward and reverse side of the DNA. Okay, now these pieces go and attach to the flow cell. So flow cell is like a glass slide, okay, which has both purple and pink. You can see purple, blue here, okay, various adapters. Okay, so now the purple side of the DNA will go and bind to the purple side, and blue side will go and bind to the blue side, okay, and this way. Okay, so like here they have bound, okay, and then for each piece of DNA you have amplified that into, okay, multiple pieces, and then, okay, so this is basically called amplification on the flow cell or bridge amplification step. Okay, so if we go into details of this, so like this was one piece of DNA that was bound here, okay, then by using fluidics you will tilt this DNA and this will form a bridge with the adjacent pink adapter which is there. Okay, so like here you can see the pink side was bound here, it tilted and found the adjacent purple adapter here, and now this purple adapter also act as a primer to this, and now putting a polymerase, it will make a keep inserting DNA the nucleotides and make a copy of it. Okay, so like this bridge is now amplified into two copies. Okay, so this will be dissociated into two copies. Okay, you can see, which are basically opposite of each other, right, because this was earlier tilted here, okay, and then right like this. Okay, so this way you will amplify each and every fragment that you have on the flow cell, right, into a certain number of fragments per piece, okay, or certain number of copies per piece, which is about 500 to 1000, right. So you can see that for every position in the flow cell you have a copy of the same fragment at that position, right. Okay, so now we have sufficient number of copies at one position, and you can see that it contains both purple and pink DNA, okay, and purple and pink are basically, you can say, reverse complement of each other. So we have to get rid of any one color. So for example, here we have removed the blue ones or the purple pink ones, okay, and got only a single color, right. Okay, so this way we are not, otherwise if I sequence this I will get confused because I will get signal from both forward and reverse fragment. Okay, so now here all the DNA are in a one direction or forward direction, right. Okay, which have all purple adapters on the top and blue ones at the bottom, right. So now each piece will have the same orientation of or arrangement of nucleotides, and when I start reading from one side I'll get a signal for example A is giving signal here. So all the pieces will give the signal A once the polymerization of or the DNA polymerase is acting there, right, and this is called sequencing cycle. So let's see here. So now you can see that each position contains multiple copies of the same DNA fragment, right. So when I start the sequencing, how I start for this fragment basically, okay, so I put a primer, okay, and then one nucleotide got inserted, okay, polymerase was there, right. So one nucleotide got inserted, okay. So for each piece of DNA, it will be the same nucleotide which got inserted. For example, A was here, so A will be here in each piece. Okay, so now together there will be say 500 copies of A's. Okay, there were 500 copies of that DNA, so 500 A's were there. So 500 A's will give me one signal of A. Okay, like you can imagine, like if A is giving red color light, so there are 500 bulbs of red color light which are glowing together. So I know that okay, A is there in this cluster. Similarly, in this cluster there could be some other nucleotide, it could be say T. So all position T will get inserted and give me another say green color bulb light, right? Okay, so this way I will know that okay, in this position in a cluster it was A was giving a red light, so it was A. Okay, and this position was T. Okay, and this position was C, and this position was G. Okay, so for 4 different clusters, if I see on the flow cell, I will know that for the start of sequencing or the first cycle or for the insertion of a particular nucleotide, what nucleotide got inserted at that position. Okay, and that I get by the laser excitation. Okay, so this way I will be doing massively parallel sequencing throughout the flow cell. Okay, there you can imagine there are more than 100 million fragments which are attached on this flow cell, and they are giving like any of the 4 color light at where the okay, so you can see that imagine a field where people are glowing bulbs, they are standing in clusters and glowing bulb. So you can say that sometimes they will glow A, sometimes B, sometimes C, sometimes C. Okay, so and their position is fixed, right? So it is not that this cluster will move its position. So in XY coordinates or in the 2 dimension, this position will be fixed. So whatever light I am getting from here at the different cycles will be dependent upon what nucleotide got inserted into that DNA. Okay, so this way I'll keep on inserting the DNA. Okay, reading the fluorescence for T, removing the blocker because if you remember it was a nucleotide that had a reversible blocker. So I'll remove the blocker so that the OH groups becomes free for accepting or going and so that the polymerase can add another nucleotide to that nucleotide. Okay, the chain can grow. Okay, then G comes, again it will be paused, signal is read, and then the next nucleotide is attached after removing the blocker from G, right? And in between, because there will be several nucleotides which do not get incorporated because we are actually throwing all the 4 nucleotides together okay, in this, right? And any one of those will get inserted into here, and we are always throwing the nucleotides in excess, so the remaining ones are removed or washed out every time, otherwise they will remain there and they will interfere with the signaling. Okay, so this way we'll do the cycle 3, cycle 4 and cycle. So basically these one cycle means incorporation of 1 nucleotide, right? So if I say that the sequencing by synthesis or the Illumina sequencing can give 150 base pair length read, so that means I am doing 150 cycles here. Okay, and each cycle includes incorporation of a nucleotide, removal of the unincorporated nucleotides, reading the signal, removing the blocker, and then proceeding with the next cycle. Okay, so now on the flow cell I will know that okay, from this cluster gave me A signal, this cluster gave me C signal, T and D. So I will know that okay, this part is giving me G signal, this is A, T and C, and this way I can keep on reading. Okay, for this cluster it gave me like T here, and then it gave me A, it again gave me A and G, because the position is fixed, you can see at this position, and then it gave me T. So the sequence and in this cluster is at this position is T, A, G, T, and similarly at this position it is if I start from here, it is A, then G, then C, then C, and then G. Okay, so this way I can construct the sequence from each and every cluster, and I can go So whatever number of cycles the flow cell was, like it was say 150 cycles, so 150 base pair, I will know that what are the 150 base pairs at this of this fragment of DNA. Okay, so you can see that actually the signal is coming like this. You can, okay, so you will know that, okay, this position 1 2 3 4 5 6 7 8, so 9 base pairs which are in this strand, piece of DNA are 1 2 3 4 5 6 7 8 9. So I will know that what 9 nucleotides are there, okay, and continue like this. Okay, and you can see that if you are reading signal like this, you need a camera which is of very high resolution, because you can see that fluorescence from neighboring clusters also may get mixed up. So a very good computational software is required to separate or resolve this. Okay, because you can see that in this cluster it could be like yellow color, this is red, this is blue, and there could be very close to each other. Sometimes there could be two blues which are very close to each other, right? And you have to separate them or resolve that, okay, these are two different blues but not a single blue. Okay, and so you need a very good camera and you need a very good software which can resolve, which can read this image and resolve it. Okay, so basically what we are doing is, from a flow cell, okay, there are different clusters, right, and we are taking picture of each region. So okay, not every cluster but a bundle of clusters, like for example this picture contain 4 clusters, okay, and that way we are taking picture from the entire flow cell. So we are dealing with like millions of pictures that are taken for each cycle, right, and then reading that picture to get the sequence of DNA. For example, here the sequence is read like this, and this one, okay. So these are 3 or 4 different clusters which are giving me different signals and different based on these DNA sequence that they contain, and this way I can resolve the DNA sequence. Okay, so as I mentioned, like it contains a blocker. So OH group is there, but it's a reversible blocker, okay, 3' OH group, and similarly there is a fluorophore attached at one corner. So both of these are removed, okay, so that the OH groups become free to accept the next nucleotide, and this blocker is also removed so that the fluorescent signal that was there initially do not interfere with the next cycle or the next step or the next nucleotide. And that is why this reversible terminator chemistry is very important, and which actually leads to the sequencing by synthesis, right. Okay, again, like this is the open instrument, and we have removed the covers from here, and you can see that it is a complicated one, right, and this is a picture that comes out of the sequencer. The flow cell is placed here, and there is a camera, there is a laser source which is exciting this flow cell, and this camera is reading, taking picture. Okay, so what is a flow cell? So you can see like this is a flow cell which is like a glass slide which contains 8 different lanes. Okay, these are 1 2 3 4 5 6 7 8 lanes. Okay, and each lane and basically in this there is a place here you will put your DNA library. Okay, and the requirement of initial to prepare the library is about 1 microgram of DNA. Even 1 or 10 nanogram is also sufficient for the highly sensitive kits, right. And okay, so now this is a flow cell, it has 8 lanes. Okay, so each lane, if you see like this lane is actually like this, okay, which is divided into 3 columns. Okay, and each column has various tiles, right. So there are 100 tiles per column, so basically 3 columns into 100 tiles. So basically you can see that there are 300 tiles, and one camera, maybe if it is taking picture of one tile, okay, and one tile is like this, okay, so it is taking picture of the fragments which are present in that tile. Okay, so this way the camera will keep on moving from here to here to here, here like that, and it will be taking pictures for all the tiles. Okay, and then the software will be reading those pictures and segregating the various clusters which are there, which are giving the signals, and also the deciphering their sequences. Okay, and after you have done with the sequencing, you have to do the computation analysis, you have to see the forward and reverse reads, you have to align that with the reference, or do the de novo genome assembly, right, and get the initial DNA that you took, that you want to get back by assembling those pieces together. And again, for various purposes, there are different type of sequencers for clinical purposes, for large scale sequencing, for like common research labs. Okay, so like this is a very small sequencer, mini-seq, which can serve the various clinical labs. MySeq and NextSeq are very good for research labs. HiSeq and NovoSeq are basically meant for very large sequencing centers which do sequencing all the time, 24 hours, or even the companies that where you outsource your sequencing, and they produce data for you. Okay, and each of those machines have their own output. For example, iSeq can produce 1.2 GB, which is good for very small project or clinical purposes, because you don't want to put, you don't have too many samples, and you don't want to, you know, waste consumables also. So getting 1.2 GB there, but if you have a lot of samples to do, you can go for like NovoSeq, which can give almost 6 terabyte, or okay, 6 trillion base pairs, okay, together. Okay, and then in between you have several range of sequences like NextSeq, which is good for the research lab, which can give up to 120 billion base pairs in one run of say 24 hours. Okay, and the sequencing, the read length will vary from say 75 base pair to 150 base pair to 250 base pair also. Okay, depending upon the various requirements, and it could be the paired end sequencing, or it could be the single end. Again, based on the requirement, the user can decide. And again, you can do multiplexing, because you don't need to, for example, it can give 6 terabyte of DNA it can read 6 terabyte of DNA sequence. So you don't need to put a single genome here. You can also have thousands of genomes libraries made, and put the barcodes with that, so that you can put multiple samples together, okay, which is called multiplexing, and then you can later resolve those barcodes and get the you know, various sequencing for the various fragments that you have put. Okay, so Illumina is a very good sequencing technology, and it is free from errors, and there are so many advantages. For example, it is massively parallel. Okay, you can do millions of pieces together. Okay, you don't need to do like gel electrophoresis that you do in the Sanger one. Okay, speed is very high, massively parallel, very low cost, and it is decreasing every day. Okay, it is becoming more and more efficient and cost effective. Okay, very high throughput and very low error rate. So all these things are in favor of Illumina sequencing, and that is why it has stood the test of time and surviving from, I think, last like 15 to 20 years, and it is becoming better and better. But some of the limitations are that it produces short read lengths, as I mentioned, that 75 base pair, 150 base pair, 250 base pair, 300 base pair, okay, which poses a challenge for assembling very large genomes back, and there will always be missing pieces in the genomes. So that is one disadvantage of this sequencing. And when you have so many pieces also, you need very you know, like highly parallel computing systems for like doing the assembly and you know, further computation analysis, which is also a computational burden, right? And PCR amplification is required sometimes, otherwise it can also be done on the flow cell, right? And basically it is not meant for a single piece of DNA, right? Like in Sanger, you can give one piece of DNA. For example, I amplify a 16S ribosomal RNA gene and give it for sequencing. But I cannot do a single piece of sequencing here, because you know, I cannot run a like sequencer partially or the flow cell partially. The entire flow cell will be run together. So even if I am doing one piece of DNA or millions of pieces of DNA together, the cost will be same. So it is not meant for single pieces of sequencing, but for massively parallel sequencing. So what we have learned today is the principle and concepts of two very important sequencing technologies, that is Sanger and Illumina. And Sanger is first generation, whereas Illumina is second generation sequencing technology. And in the future classes you will see the other latest sequencing methods also, which are very important because these methods actually are generating the big data of biology, which we are using for the computational genomics and for various computational purposes. Okay, so thank you and see you in the next class. Thank you.